Testosterone, a steroid hormone predominantly synthesized in the testes of males and to a lesser extent the ovaries of females, and in the adrenal glands of both sexes. Testosterone is an important member of the androgen class. Androgens are crucial in the development and sustenance of male characteristics. Testosterone controls a plethora of physiological processes such as maturing reproductive tissue, regulating muscle mass, and regulating energy levels. As previously stated, chemically, testosterone is a steroid hormone distinguished by its molecular structure comprised of four interconnected carbon rings. This structure renders testosterone hydrophobic. As a result, testosterone's transportation through the bloodstream and its interaction with target cells and receptors, notably the androgen receptor protein located within the cytoplasm or nucleus of cells, are influenced by testosterone's hydrophobic properties, the same properties that make testosterone able to diffuse through the semipermeable lipid membrane, making it able to bond to the androgen receptor protein. The purpose of androgen receptors is binding to androgen hormones such as testosterone, and in turn regulating gene expression. Androgen receptors play a key role in multiple physiological processes such as the development of male sexual characteristics, promoting growth, and repairing muscle tissue. There are a couple advantages of using the androgen receptor protein, one of which being specificity. Androgen receptor proteins exhibit specificity in their binding to testosterone molecules, ensuring that the hormonal signals are directed only to cells possessing these receptors. This specificity helps to regulate the target tissue and prevent unintended effects on other tissues. Another advantage, as stated earlier, is regulation of gene expression upon binding with testosterone. Androgen receptor proteins can modulate gene expression, leading to the production of specific proteins that regulate various physiological processes. Androgen receptor proteins are also important in regulating the differentiation and development of reproductive tissues, secondary sexual characteristics, and other androgen responsive tissues in both males and females. This ensures proper development and function of these tissues. Feedback regulation. Androgen receptor proteins participate in feedback loops that regulate the production and release of testosterone and other androgen hormones. This helps maintain hormonal balance within the body. Although there are advantages of the androgen receptor protein, it comes with disadvantages as well. While androgen receptor proteins provide specificity in hormone action, their presence in various tissues can also lead to tissue-specific effects of androgens. In some cases, this can result in undesirable effects such as acne, hair loss, or prostate cancer. Dysregulation of androgen receptor activity or expression can lead to hormonal imbalances, contributing to conditions such as androgen insensitivity syndrome or androgen receptor related cancers. While androgen receptor proteins are crucial for normal physiological functions, they can also serve as targets for pharmacological interventions. Drugs that modulate androgen receptor activity are used in the treatment of conditions such as prostate cancer, but they may also have side effects related to androgen receptor function. Overall, while androgen receptor proteins play essential roles in mediating the effects of testosterone, their function and regulation must be carefully balanced to avoid adverse effects and maintain proper physiological function. The group of receptor proteins exemplified by the androgen receptor functions through a process reliant on ligand receptor binding and gene expression regulation. Initially, these receptors are mainly located within the cytoplasm of target cells in an inactive state. Upon entering their respective ligands, such as testosterone, these receptors undergo a conformational change, transitioning into an activated state. This activation enables them to translocate into the nucleus where they bind to specific DNA sequences. This binding event serves as a switch regulating the transcriptional activity of these genes. Depending on the cellular context and presence of co-regulatory proteins, the ligand-bound receptor complex can either enhance or inhibit the transcription of target genes, thereby eliciting cellular responses. The stimulus for reaction is testosterone. The specific signaling pathway is testosterone, which primarily is responsible for activating the androgen receptor. Speaking of the androgen receptor, it is the key receptor in this pathway. As far as molecules that are involved in the intracellular response, transport proteins are one example. They control the translocation of the androgen receptor. Another key molecule is the regulatory proteins. These proteins maintain hormonal balance inside of the body. Therefore, they can regulate the expression of the androgen receptor in an effort to regulate testosterone levels in the body. Androgen response elements are also key molecules in the signaling pathway, as they serve as binding sites for the androgen receptor. In the androgen receptor signaling pathway, the sequence of events unfolds as follows. Initially, testosterone, the specific signaling molecule, diffuses through the cell membrane and binds to the androgen receptor. This binding triggers a change in the receptor leading to its activation. Then, the activated androgen receptor complex translocates from the cytoplasm into the nucleus of the cell, where it binds to specific DNA sequences located in the regulatory regions of target genes. Through this binding, the androgen receptor modulates the transcription of these genes, either promoting or inhibiting their expression based on the cellular context and the presence of co-regulatory proteins. The altered expression of target genes controlled by the androgen receptor signaling induces various cellular responses, such as the synthesis of specific proteins, alterations in cell proliferation or differentiation, and adjustments in metabolic pathways. Finally, the pathway is subject to feedback regulation mechanisms aimed at maintaining hormonal imbalance within the body, involving the regulation of androgen receptor expression and the activity of enzymes implicated in androgen biosynthesis and metabolism. The intended response of the cell is gene expression regulation. The androgen receptor complex regulates the transcription of target areas, leading to the synthesis of specific proteins involved in various cellular processes. These processes may include cell growth, differentiation, metabolism, and immune function, particularly pertaining to male reproductive and sexual characteristics. The termination of the androgen receptor signaling pathway involves a combination of processes: degradation mechanisms, feedback regulation, and modulation of the receptor activity, all aimed at restoring cellular homeostasis and preventing excessive signaling. The general class of antagonists in the testosterone signaling pathway is called anti-androgens, which are exogenous. They are created from external sources. They interfere with the receptor signal, thereby antagonizing testosterone's effects. Furthermore, anti-androgens can be categorized into two classes: competitive anti-androgens and non-competitive anti-androgens. Flutamide is an example of a competitive anti-androgen. It blocks receptor binding of the androgens and slows down transcriptional activity. In general, competitive anti-androgens compete with testosterone for binding to the androgen receptor. By binding to the receptor with higher affinity than testosterone, competitive anti-androgens effectively block the activation of the receptor by testosterone, inhibiting downstream signaling pathways. Non-competitive anti-androgens, such as cyperterone acetate, work through mechanisms independent of androgen binding. Instead, they interfere with androgen receptor signaling by inhibiting various steps in the signaling cascade, such as coactivator recruitment, or ultimately preventing the transcriptional activation of target genes. This response to testosterone signaling is altered or changed by anti-androgens primarily through inhibition of androgen receptor activation and downstream signaling pathways. That's all. Thank you for listening.